The overall goal of this procedure is to transfer previously isolated mitochondria from donor cells such as mesenchymal stem cells to target cells such as glioblastoma stem cells.
This procedure can help answer key questions in the metabolism field, such as the metabolic and biological effects of the mitochondria that are transferred spontaneously between cells by processes such as tunneling nanotubes or microvesicles. The main advantage of this technique is that the effects of the mitochondria can be assessed independently of other factors present in the microenvironment, such as cytokines or metabolites, and also of other compounds that can also be trafficking in the tunneling nanotubes.
For imaging purposes, MSC mitochondria and JEC can be labeled with vital dyes before the mitochondria transfer by MitoCeption.
To begin, after pre-labeling GSCs with vital dyes according to the text protocol, collect GSC neurospheres in a 50 ml tube by centrifugation at 270 times G at 20 degrees Celsius for 5 minutes. Use 5 ml of HBSS to wash the cells and spin the tube again. Aspirate the supernatant and with 100 microliters of 0.25% trypsin EDTA, gently resuspend the GSC pellet. Then incubate the cells at 37 degrees Celsius for 3 minutes. Next, add 10 microliters of 20 millimolar calcium chloride and 2 microliters of 10 mg per milliliter of DNase-1 to the tube.